Okay, cool. Now we are live. And hello, everyone, and thank you for joining us to this ISCB Student Council International Society for Computational Biology webinars. I would like to remind you, if you want to know about the upcoming webinars or how you can present your research and be part of this amazing initiative, you can follow us on Twitter as at ISCB-SC. And today we have an interesting talk by Kavisha Dilshani from University of Perania in Sri Lanka. And her presentation is titled Identification of Alzheimer Disease Using Gene Expression Data. So thank you, Kavisha, and the floor is yours. Please go ahead.
Okay, thank you. Work on detection of novel biomarker genes of Alzheimer's disease using gene expression data. So this was actually a collective work done by the Department of Computer Engineering, University of Perera in Sri Lanka. And today I will be presenting on the team, on behalf of the whole team. So before moving on to what we did, let me introduce to you like what Alzheimer's disease is. So it's the most common form of dementia, and a successful medication or a cure for the disease is not found yet. So the interesting thing is that the disease has been known for around more than 100 years, but still the scientists and researchers are struggling to find a successful medication or a cure. So the main reason behind not being able to find a successful cure for the disease is that the scientists are not exactly sure of what the real reason or the cause behind Alzheimer's disease. So there are many areas of research that are trying to identify the hidden cause for the disease, and one such approach is trying to explore the underlying genetic structure of the disease. So, in that case, also, there are many ways that scientists and researchers are using, even for today, to explore whether there are any biomarker genes that could have a potential risk for Alzheimer's disease. So one such form of data that we can use to extract information regarding those biomarker genes is gene expression data. So let me introduce to you like what gene expression data is in really simple terms. So gene expression is the process of converting the information encoded in a DNA of a gene to functional protein. So the state of gene expression at a given time is an indication of the state of the cell at the given time. So by comparing two or more samples in two different conditions, we can like extract meaningful information as of to what are the genes that have been like differentially expressed between the two samples. So there are various techniques that are being used to extract gene expression data, and the main problem with gene expression data is usually the data set is really huge and contains lots of data that couldn't be like processed using normal conventional methods. So complex knowledge on statistical methods and also knowledge on programming skills and computational methods are needed if we are to extract meaningful information. So what we have done in our work is that we have used a machine learning approach in extracting the genes that we can predict as the most impactful gene for the Alzheimer's disease. So what exactly is machine learning? So according to a definition given, machine learning is an application of artificial intelligence. So it provides the system's ability to automatically learn and improve from experience. So usually what we do is as programmers and computer scientists, they usually write the code and program to get some expected results. So, but in machine learning, the difference is that it focuses on the development of computer programs that can access data and learn by themselves without being explicitly programmed. So that's the specialty in machine learning. So if we are to give a brief overview of what we have actually done. So first we collected a data set. So the data set was collected from NCBI Gene Expression Omnibus. So the exact data set that we used was GSC 5281. So it contained samples information from 161 samples. So out of those 161, 87 samples belong to patients that have been detected with Alzheimer's disease, and 74 are used as control samples to compare whether there's a difference between the samples from Alzheimer's disease and the normal patients. So after that, the our main focus is, since the initial data set is really huge and contains data with lots of noise, like that means like data that can like have have differences and lead you in the wrong way if not handled properly. So we have to first apply some data pre-processing techniques. So in those pre-processing techniques, what we do is we like clean the data and prepare the data so that we can derive useful information later on. So in the initial steps, so in the very first step that we downloaded the dataset, it contains around 54,000 features. Features means information uh, about probiotics in microarray technology, where the gene expression data was extracted. So handling around 54,000 features from a small sample like 161 is really difficult using normal methods. So that's why first we had to like map the probiotics to gene symbols, and also we did some things to like, there were some missing values in the middle, so we had to consider about them also, and then we had to do what we call as normalizing to like get the data into a one range. And after doing those things, we filtered that 54 that features around 54,000 to a feature count of around 22,000. So even if we reduced it to 22,000, like it, even those that count is a really large count when we need to get some meaningful data. So as the next step, we applied a method called univariate selection, where you can like filter out and obtain, like it gives a score where uh, so before applying univariate selection, we applied some statistical methods using p-values and fold chain. So those three methods together, like univariate selection, p-value, and the fold chain, uh, gives you a score on how to get the most differentially expressed genes out of those 22,000. So after applying those three methods, we were able to narrow down our feature set to around 200. So after that, 200 was the main difference in our work. So there are methods called features, what we call feature selection in machine learning. So in that, we apply some special methods uh, to get their scores based on the like, it gives an indication of the importance to the targeted output. So by using those feature selection method, we actually selected three feature selection methods called principal component analysis, and also from scores from a method called uh, extra tree classifier and random forest. So from those three methods, we selected 50 genes from each method. So after that, we'll see what happened. So after we selected the best 50 genes from those three methods, you can see that some genes were found by two methods, or in some cases, so in this case it's eight. So eight genes were found by all the three methods. So what we did was we plot we can get get the accuracy of the selected genes by plotting the accuracies with what we call as classifiers. So classifiers are used to, like, it what it basically does is it classifies the our results into two samples, and like by comparing the results that the in this case we call that a model. So comparing the results that is given by the model and the actual results, we can get an estimation of the accuracy. So if we plot the accuracy for the genes found only by PCA random forest and extra tree classifier, and also if we get the accuracy for that overlap set of 8 genes, we can see that there's a huge like in the graph in the right side, you can see that there's a significant improvement in the set with the overlap set of genes. So so from this result we can derive that rather than like selecting only the genes are found by one method alone, by combining those three methods, we get a significant improvement in the accuracy of the final results. So in this, it shows the accuracies without the overlap set, and another thing that we focused on our research was as you might already know, like genes are usually highly correlated. So if we directly apply machine learning techniques or statistical and computational methods, it won't take into account about that fact. So what it does is like it just mathematically computes the scores without considering any fact about the correlation. So in here, what we focused on was deriving the best set of biomarker genes. So for that, we didn't need to get the redundant data. So to minimize the redundancy and to get the genes that will give novel information only, we focused on reducing the redundancy aspect in our selected features as much as possible. So for that, we plotted what we call as a correlation matrix. So as you can see in the diagrams, you can see like in simple terms, if the color variation is high, that means the genes are less correlated. So like the similar colors show similar genes in kind of a way. So in here also you can see like in out of these three methods, you can see a significant difference in the genes found from the PCA. PCA means principal component analysis method. So out of these three methods, we thought of selecting PCA in moving further for our analysis. So this graph explains our point further. So you can say that this graph in the y-axis, it shows the correlated features as a percentage, and in the x-axis it only shows the correlation values. So that means like in PCA it's shown in the blue color, like until up to around 0.86, none of the features have been correlated. Like in the at the same point, if you get like random forest, you can see like more than 50%, like it's almost around 60%, 60% of the genes have been correlated. So this graph again validates our point that PCA is a better estimator or a better feature selection method in when we focused on reducing the redundancy aspect. So from combining those, uh, so in the earlier steps we got around 8 genes, and from considering PCA and with also by training the set with the classifiers that I told earlier, we found around we found another 6 genes from considering PCA alone. So those two sets of genes combined, like the OLAF set and the genes found only by considering the PCA, uh, we we got an accuracy of so these are the values of the accuracies when tested with 3 classifiers. So you can see that in all 3 classifiers the accuracies are in a high range. So this value is like a bit when we compare it to the related literature and the current research, this value is an improvement and shows some promising results. And also in like since the data set, the sample size of the data set was small, we cannot only rely on the accuracy. So to ensure that our results were correct, we also tested for the measures of sensitivity and specificity. And even in those results, you can see that they also give good results. So making it clear that our method and the predicted results can be used for further exploration. Uh, so, uh, so by combining those results, we were able to select 14 genes. So the gene symbols are shown here, and you can also see the correlation between how those genes are selected. And also, we we found that our out of those 14 selected genes, some genes have already been identified and validated by other biological sources that are related to Alzheimer's disease and have a high impact on the disease. So with those validations and with the new results that we found and with the correlation between those newly found genes, we can predict that these 14 genes also might have some impact on Alzheimer's disease. So as the conclusion, from our work, we found 14 other genes that could potentially have a risk for Alzheimer's disease. And also, the one of the key findings on our research was that principal component analysis is a better approach for minimizing redundancy. And also, like in this experiment, we have only used data sets collected from microarray technology. So, we got some feedbacks from people related to biological aspects also, like that rather than using microarray technology, it can be like have a more impact or a more usefulness if we can use or integrate the next generation sequencing data to this approach as well. So that is one area that is open for more research in this work. And so that's all about our work. And as I said earlier, so this was a collective work done by the Department of Computer Engineering, University of Peradeniya, and the contributors were Shehan Perera, Kavisha Hevage, Chamara Gunaratna, and Professor Roshan Ragal, Dr. Damayanti Herat, and Dr. Rajita Navaratna. And also one thing I have to mention is that this work has already been accepted and published at MOCON 2020, the Moratuwa Engineering conference. And so this was a brief abstract on what we did. So the detailed research will be published in IEEE Explore in the near future. Uh, and yeah, those are the references that we used, and thank you.
Thank you, Kavisha, for your interesting talk. So now we have time for questions from the attendees. So let's wait a few minutes. So, but in the meantime, I can to comment something about your work. Have you thinking to test your algorithm in different stages of diseases? I mean, because maybe in complex diseases, you are not going to have always
Mhm.
the same biomarkers. Maybe in different stages of the diseases, you can find different genes. Maybe that will be nice to test with
Mhm.
RNA-seq data or using another disease as a model of study.
Yes.
I don't know if you think to perform this as a perspective of what are your what are the perspectives of your work, just in terms of change the data set or the kind of data, or maybe to propose another kind of modifications.
Yes, so like that was also an issue that we faced, like in Alzheimer's disease, especially like there are various stages, and in the data set, like they contain some metadata. So in some data sets, like they have three categories, like one for normal patients and the other in the mild cognitive impairment state, which is kind of the middle state, and also in the Alzheimer's disease state. So in what we have focused is only the normal state and the Alzheimer's disease state. So we have excluded the mild cognitive impairment state. So it will be like really useful and better if we can
Hmm.
integrate that also. But in what we have done so far, we have not included that.
Okay, yeah, that would be great. Okay, there is, I think then there are no questions. But once again, thank you, Kavisha, for sharing with us your research, and thank you.
Okay, thank you very much.
Bye bye.